understand glycolysis. Such a high yield topic, I think, right? Glycolysis. Okay, that's our topic of choice today. Nice topic. I really like it. Very simple topic. So, to understand glycolysis, first is that I'm gonna build your concept about before what happens glycolysis, right? Before what happened your glycolysis, right? You mean we need to know, all right, what's happening before glycolysis. Then we will understand why glycolysis even exists. Then we know why, and just overall glycolysis thing, like this overall, right? And then we want to understand the reaction, glycolysis reaction itself, okay? And in this reaction we have around 10 steps, okay? We have around 10 steps. So from step 1 to 3, these are irreversible steps. We cannot reverse them. I mean, the enzymes cannot reverse them. And from step 4th to 10, these are quite reversible steps. So we will understand differently, right? And in the end, when we are done with overall reaction, we'll understand why only step 1, 3, and 10 are irreversible, okay? Even 10 is irreversible, okay? And we'll understand what are the regulation, right? Who is taking care of this whole thing, whole operation? And then we want to understand clinical relevance of this. Clinical relevance, like why it's important, like what's the deficiency, blah blah blah. And then in the last part, when we understood clinical, I will summarize everything. I will integrate, okay? So integration. Okay. So that's our goal. That's how we're gonna understand glycolysis, okay? I will not assume that you know anything about glycolysis. I will act as if you don't know anything, okay? So bring your coffees, bring your chai, bring your water, whatever. It's gonna be a long video because I'm gonna break down everything, okay? Okay, so let's start with the first part. What is happening even before glycolysis, right? Okay, so listen to me closely. Before we learn glycolysis, we should not memorize anything, okay? Right? We don't need to memorize this whole reaction because memorization is not gonna take you far. Instead, what we are gonna do is we're gonna ask better questions, right? The better questions we ask about this reaction, the better we understand it, right? Now, I feel to understand anything in science is to ask why and why not. These two questions are very important. So first question I want to ask is why. Second question I want to ask is in which specific conditions does this reaction fails, okay? So that's important. Now I'm going to ask a very simple question right now. Why does glycolysis exist at all, right? This is the most logical and the basic question, right? Now I'm going to ask this question. Why is this even happening, right? Now to answer this, I want to build slowly, right? Now I want you to imagine that you are a cell, yourself. You are this cell, right? Now what's your biggest problem right now? What do you think? It's not glucose. It's not oxygen. It's not ATP as well. Your biggest problem right now as a cell is that you need energy for every second to stay alive. Now this is an assumption, right? I'm telling you, you need that. You must be thinking, but Shashwat says that you don't need to assume anything, right? So I'm going to tell you, I'm going to prove this to you that you need energy every second, right? Let's prove it. Now, what does a cell spend energy on overall? So when you're sleeping, every cell is busy. The cell is not sleeping. You are sleeping. Your brain is sleeping, right? A cell must constantly put sodium out, right? So first thing a cell needs to do is put sodium out, then pump potassium in, right? You must be thinking, why? It is to maintain an electronegative gradient, okay? So it is to maintain membrane potential. Then cell needs to repair what? Repair the DNA that was damaged during its replication phase, right? Then it needs to contract if it's a muscle, right? Then it needs to even fire action potential if it's a neuron, right? Because you are dreaming. How does dreaming happens? By action potential, right? Then it needs to divide even because you sleep, you build your muscles, right? You need to transport molecules. I mean, you get me, right? So question is, can any of this happen without energy, right? No. Life will stop if you don't have constant supply of energy. So we need a constant supply of what? Of energy. And that sucks, honestly, because you can't eat every time, You're not eating all the time. You just eat every 8 hours, right? So, next thing I want to ask is, why can't cells simply stop using energy? Well, if they stop using energy, the cell will fail. Cell will die, right? Everything will stop. The deeper meaning of this constant supply of energy, why we need constant supply of energy, is to hold the second law of thermodynamics. Now you're saying, oh, what is this thermodynamics? Why is he mentioning this thermodynamics in glycolysis? Because I'm building your concept from fundamentals. Second law of thermodynamics says that system naturally move toward higher entropy. Now you're saying, what is higher entropy? Well, higher entropy is just higher permutation and combination that are available in a free system. In a system. Okay. For example, your room becomes messy if you don't clean it. So nature is moving towards messy room, right? And you need to spend your energy and your cognitive energy to think and to organize and to put order in your room. So messy room is entropy, high entropy, and ordered room is a less entropy. Nature always moves towards higher entropy. Okay? So without energy, the proteins will unfold, right? Ion gradient in the cell, it will disappear. Membrane will lose its organization. Eventually, the cell will die. And therefore, the cell must constantly import energy to maintain its order. So that's why we need a continuous supply of energy. This is the reason. Because we want to fight entropy, right? Simple. Now, where does this energy even comes from, right? Does the food directly powers your cell? Because I'm sure you're not eating every second, right? You only eat like every 8 hours or 6 hours, depending upon how fat you are. Okay, that's a bad thing to say. Okay, so food is what? Food is a stored chemical energy. Now, cells cannot use this piece of bread directly, right? They must convert this piece of bread into the energy, right? They must convert the food into universal currency. Now, universal energy currency. Now, what is that currency? What is that converted energy from food? So food must be converted to energy. What kind of energy? We call it ATP. Now you must be thinking, oh, I know that. I read that already. Yeah, I'm sure you did. Now, first question I want to know is why ATP? Then want to know what is ATP? right? We want to know that. Let's talk about why. Why didn't evolution use glucose, the food itself? Why didn't we use it itself, right? That's an excellent question. I mean, why even use ATP? Because it'll be just better. Just imagine you're traveling in the world and you don't have to change your currency every time when you move from USA to, I don't know, any third world country, or you move from third world country to USA. You don't have to change your currency, right? Suppose every machine just accepts maybe yen, right? Or maybe pounds. Instead, imagine, just instant. So that's the idea. ATP is a biological universal currency. Every enzyme can understand ATP. So it's a universal currency. That's what ATP is. Every cell understands it. Every cell is like, yeah, I know this. Okay, I'll accept. Now, second question is, what is ATP then? ATP stands for adenosine. Now I'm going to say triphosphate, okay? Triphosphate. Now we have two words here, adenosine plus triphosphate, right? Now what is adenosine then? Second question arises, right? Adenosine is made up of adenine, same thing that is used in your DNA, okay, plus ribose, sugar. So that's important. And this too forms adenosine. What about triphosphate, Shashwat? Well, triphosphate, these are three phosphate groups, okay? These are three phosphate groups. They are attached with each other. Conceptually, they are attached like this. That is adenosine, okay? Attached phosphate, then they are bond, and phosphate, right? Now here notice this part, right? This one, okay? Do you see this bond? Yeah. So we have these two of them, right? What is it? This squiggle, right? What the hell is this? Why I didn't just put a straight line? Because this squiggle, it represents a very high energy, right? It represents high energy phospho anhydride, okay? Remember this word, anhydride bond. So it has phosphoanhydride bond, this thing, very high energy. We'll study this high energy later because I'm building this concept step by step, okay? Just be with me. Now, thing is that you can ask me, so Shashwat, this ATP, does this has energy? Or what is it? Like, what exactly physical thing it is, right? Many books will tell you that ATP stores energy in bonds, right? Now, this is a very oversimplification, right? More accurately, when you break these ATP bonds, we get energy. But breaking these ATP bonds alone requires energy, right? So the overall hydrolysis reaction. Now, wait, hydrolysis, what is it? Hydrolysis is when we use water to break hydrogen bonds, okay? Or not hydrogen bond, just break the bonds, right? So, wait. So the overall hydrolysis reaction, we use water. We are basically water, right? We are 90% or not 90, 70%, 60% water, right? So we use this water to perform hydrolysis on ATP, right? And the overall hydrolysis reaction releases free energy like ADP and pi. And this free energy, right? This ADP and pi, the result of hydrolysis of ATP, is way more stable than ATP itself. Now this is very important, okay? So ATP, let's break down ATP. We break down ATP using hydrolysis, right? Hydrolysis. And then what are we gonna get? What's the product? Right, we spend some energy on ATP. Now the bonds are broken, right? What do we get? We get ADP. Now we get adenosine plus duo 2 phosphate plus pi, right? Now what is this? This, I say pi, wow, I'm so stupid. This is inorganic. I is inorganic phosphate, right? It's a useless, not useless. We just broke that phosphate, right? We had 3 phosphate here, right? We had 3 phosphate here. So we removed one bond, and then this is inorganic now. So that's what I'm saying. So we have inorganic phosphate, right? So ADP plus inorganic phosphate. Now this energy, right? This whole thing, this is way more stable than ATP itself, right? That's very important thing to know, right? So once we have this whole thing releases something called as free energy, this breaking of bond. So free energy, right? So cells couple this released energy to perform work. This released energy is needed for cell to keep working, right? Now you can ask me, Shashwat, why don't you just store the ATP directly, right? Why don't you store them, right? Why don't you store them? Well, we cannot actually, we can, but it's very expensive to store ATP because ATP is super duper reactive. It's unstable and it's very expensive to maintain, okay? So your body stores very little ATP, right? Just enough for only seconds if production stops, okay? Instead, body stores something called as glycogen. And body stores fat, which I have too. So these two, glycogen and fat, then can continually convert them into ATP. We can convert these glycogen and fat, right? Now you must be asking, Shashwat, why glycogen? Why? Like, why not, I don't know, something else? Right? That's a very strong question, right? Why glucose, right? Why glucose, the glycogen, glucose, right? It's body's favorite. Why not something else? Why not amino acids? Why not cholesterol, right? Why glucose? Why is it? It's because glucose is first water soluble. That's very important, right? Blood is mostly water, so glucose dissolves instantly and easily, so it can circulate to every tissue. Fat cannot, right? Fat is lipid, so fat require lipoproteins, albumins, transport systems, right? So glucose is much more easier to distribute. Second reason is that glucose can produce ATP with or without oxygen. So with or without oxygen can produce ATP, right? You can have some conditions where you don't have the required oxygen, right? So suppose oxygen suddenly disappears, right? Can cells still obtain ATP? Yes, through glycolysis. That is glycolysis. Fatty acid oxidation means when we break down these fatty acid, okay, that is simply what fatty acid oxidation is. Fatty acid oxidation cannot continue, right? Cannot happen without oxygen because it's oxidation. We need oxygen because it ultimately depends on oxidative phosphorylation. What is phosphorylation? Adding this PO4 group, that's phosphorylation, right? So this makes glycolysis essential, or maybe so much important, right? This is the main way you get energy in a hypoxic environment, right? So brain loves glucose. This is the third reason. Brain loves glucose. You must be thinking, wow, it's an assumption, Shashwat. Why don't you tell me why? Brain consumes enormous amounts of ATP, right? The brain loves glucose. Normally, glucose is the preferred fuel, right? During this starvation phase, and then when you starve yourself, you make ketone bodies, right? And ketone bodies are important too. But glucose is always the primary source of energy because glucose is simple, right? It stores these huge amount of energy, right? We have 6 carbon bonds in glucose, in this glucose molecule. So it has this enormous amount of energy, right? Reason 4, why our body loves glucose. RBCs, red blood cells. What are you saying? RBCs have no mitochondria, right? Then how they make ATP, Shashwat? Through glycolysis. That's how they glycolysis. That's how they do it, mister. Yeah. They perform glycolysis, right? And this makes glycolysis absolutely essential for RBC survival. Remember this, because many diseases depend upon this fact, this fact that RBC undergo glycolysis for ATP. Why? Because they don't have our mitochondria. Now you must be asking, why they don't have mitochondria? Because they are simpler cells. They don't have much function, right? So, Shashwat, you have been telling me a lot about glycolysis. What is glycolysis then? Now we finally are on this stage of glycolysis itself, right? So what is glycolysis, Shashwat? In simple words, glycolysis, let me write here. Simple words, glycolysis is a metabolic pathway, okay? It's a metabolic pathway, right? It's a metabolic pathway that converts what? One glucose molecule into 2 pyruvate molecules, not atoms, molecules, okay? That's important. And when it produces this 2 pyruvate, right, it produces ATP and NADH as well, okay? So again, it's the metabolic pathway that converts one glucose molecule into 2 pyruvate molecules while producing ATP and NADH. I'm gonna explain everything, don't worry. Now, what is ATP? Now, we already talked what is ATP, right? We're going to talk about NADH as well. First, before that, let's understand the metabolism itself. What is metabolism, right? We want to understand that. We don't want to skip anything because maybe you will not understand this later on. Metabolic pathway. What is metabolism? Metabolism is simply chemical reaction that is happening inside you. Simple. Take it. It's just simple words. 2 words, 2 types of metabolism we have. We have catabolism, kata, and then we have anabolism. Kata means to cut, always. Ana means build. So, catabolism is a process, is a metabolic process, right? What is catabolism? Catabolism is a process when we break these large molecules, and then once we have done that, we release a lot of energy. For example, glycolysis itself. So what is glycolysis? Glycolysis is a metabolic pathway in which we use one glucose molecule to produce 2 pyruvate molecules, ATP and NADH. Okay, so glycolysis is a catabolic pathway. What about anabolism, Shashwat? Anabolism is a pathway when we build larger molecules, and that requires energy. For example, what? For example, deposition of fat. This fat. So fat deposition, fatty acid synthesis or glycogen synthesis, these both are anabolic pathways. Okay, so that's important, right? Okay, so which one is glycolysis again, Shashwat? That is catabolism, because we are breaking glucose to produce 2-pyruvate molecule, ATP and NADH. Okay, so that's important. So 2-pyruvate molecules, ATP and NADH simply. Okay. Okay, so what else can we think about it? Why should we break it? I mean, tell me more about glycolysis, right? How to break it, what's happening, right? So why even break the molecule, right? Why nature decides to break glucose, right? Now I already told you that brain loves glucose, right? But why is that? Why brain loves glucose? Okay, because it contains what is it? Glucose. So glucose contains 6 carbon atoms. Okay, 1 2 3 4 5 6. So it has this 6 carbon atoms. Okay, now this 6 carbon atoms, inside these carbon hydrogen bonds is stored chemical potential energy. What is it? Chemical potential energy. So cell gradually extract this energy. Okay, important thing to know is that cell doesn't extract the energy, cell gradually does it. Okay, important thing is that cell doesn't just extract the energy all of a sudden, right? Not all at once. That's very important. Step by step. If all energy was released instantly by these bonds, right? If all energy was released, we would just be losing a lot of this energy as heat. So instead, enzyme capture it efficiently, right? Now must be enzyme. Now you must think that why do we need enzymes, Mister? Well, we need enzyme to work, right? I mean, okay, why enzymes? That's a good question, right? I'm just asking. I'm not giving you any sort of assumption. I'm giving you questions and then answering them, right? Then why enzymes, right? We have enzymes that break these bonds. Why enzymes? Why can't glucose simply break down by itself? We know a lot of things break down by itself in biology, right? Well, actually, glucose can. It can. But when it happens, it's far too slow. It's very uncontrollable and it's super duper inefficient. It produces a lot of heat. So enzymes solve these 3 problems, right? Slow, uncontrollable, and inefficient, right? So problem 1 is that we want to increase the speed, right? Far too slow. So why we use enzymes? That's why is there enzyme, right? Why is there enzyme in glycolysis, right? Why? It's a good question. First problem is the speed is slow speed of reaction. Second question is uncontrollable reaction. We want to control this. We want to fine tune the reaction itself. Third problem is that we want to capture this released energy as much as possible, right? So capture release energy into ATP. So we want to be efficient, basically, right? That's the thing, right? So efficient. That's important. Now, how can we be efficient? We can be efficient by storing this energy into ATP and NADH. So this is the reason why we have ATP and NADH. This is the reason. Because we are storing this energy, right? Now, you can ask me, Shashwat, you told me already about RBCs, that RBCs have glycolysis, right? So where is this glycolysis even happening? Glycolysis always happens in a cytosol, okay? So glycolysis happens in a cytosol. You can ask me now, that Shashwat, why not mitochondria? Why not nucleus? Why not lysosome? I mean, everything, a lot of stuff is happening in mitochondria, right? Why not this also happens in, mitochondria itself? Because everything in glycolysis occurs in cytoplasm. Now, why? Because it's a big because glycolysis is one of the oldest metabolic pathway. It is older than mitochondria. Glycolysis is older than mitochondric pathway. Okay. What do I mean by this? Yeah. What exactly I'm blah 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 blah. What is it I'm saying? Evolutionarily, glycolysis is one of the oldest pathway, right? Early cells had no mitochondria. Now it's a theory. I'm not sure if it's correct or not, but I mean I read that. So early cells had no mitochondria, yet they needed ATP, right? So therefore glycolysis evolved in cytosol before mitochondria even became a part of our cells. A part of which cells? Eukaryotic cells. This is the reason even today cell without mitochondria, that are your which cells? RBCs, right? Cells without mitochondria, that are your RBCs, can still generate ATP using glycolysis. That's the reason why RBCs prefer glycolysis, right? So gave you a good introduction, right, to glycolysis. We just talked about why before glycolysis, right? Now, one thing I wanna think is that when does this glycolysis becomes essential, right? When does it happen? Which cells absolutely depends upon glycolysis? Again, there are these RBCs, because RBCs don't have mitochondria. If RBCs don't have mitochondria, they will not have any TCA cycle. What is TCA cycle? We'll talk about it in the next video, or I don't know, in future video. TCA cycle, very simple. TCA cycle, basically we are producing ATP. Just think about it like that, right? So no oxidative phosphorus. Okay, so no TCA cycle, no mitochondria, no powerhouse. Then what will happen to RBC? They're gonna die. So that's why RBCs depend upon glycolysis, right? What about other parts? What about other cells that love glycolysis? What is it? Then we have something called as skeleton tissues. Skeletal tissues or skeletal muscles, right? So oxygen delivery can become temporarily inefficient when you're working out. So what happens in this hypoxic phase, in this temporarily anaerobic phase? So skeletal muscles also use under hypoxic or anaerobic conditions. Okay, so anaerobic conditions, right? What about that? We have ischemic tissues, right? Ischemic tissues reduce oxygen, greater dependence on glycolysis, right? And the last, I think, cancer cells. Cancer cells thrive on glycolysis. Even in the presence of oxygen, many cancer cells increase glycolysis. This assumption or fact, whatever you want to call it, is called Warburg effect. We will study this. Don't worry, I will cover this as well. So in this whole video, in this first video where I discussed before glycolysis, I talked about ATP is the cell's immediate currency, right? I talked that glucose is a major fuel because it is water soluble. It can generate ATP with or without oxygen. Glycolysis is a catabolic pathway. RBC depends entirely upon glycolysis, right? We talked about this. And I talked that enzymes allow the cell to extract energy more efficiently and in a faster way, right? That is important. Okay, clinical thing that we can understand is that we have an enzyme called pyruvate kinase, right? Deficiency of this enzyme that we call pyruvate kinase can cause ATP depletion in these RBCs, because pyruvate kinase is important in glycolysis, right? So just know that. Just know that. So imagine what's going to happen if somehow your glycolysis fails in RBCs. What will happen? What do you think? What will happen? We're going to have lysis of these RBCs. If we have lysis of these RBCs, what is lysis? Just cell death. If we have cell death of these RBCs, what's going to happen? We're going to have hemolysis, simple term. So if we have something which is called as pyruvate kinase deficiency, we're gonna have hemolysis. Right? Simple. So that's thing. During myocardial ischemia, right here, right? During myocardial ischemia, glycolysis becomes important, right? So that's very important right there. Let me just underline that part. So that's important. In exercising, skeletal muscles are in this exercising, we have this increased glycolysis, right? It helps to meet ATP demand when oxidative metabolism fails, right? When we don't have enough oxygen, right? So I just added one little concept here, that pyruvate kinase. I'm gonna discuss this pyruvate kinase story and how it leads to hemolysis in the next coming videos, right? So in this video, which is 36 minutes long or 37, we discussed about before glycolysis. Okay, we discussed, I just had a little bit of foundation, because to build a concept like really deeply, we want to know what's really happening and why it's happening, right? So that's the concept. So life requires energy, right? Simple. ATP is the universal energy currency. ATP stores a lot of energy, but ATP stores are very small because they are super duper reactive. So that's why body stores energy as glycogen and fat, right? You break glucose, you get energy. Glucose has these bonds, right? And then to break these bonds, you need enzymes, right? And you need essentially energy all the time, right? So this glycolysis happens in cytosol. Why cytosol? Because it's older than mitochondria. So that's why in cytosol. So in next video, I'm gonna talk about glycolytic pathway as one story, okay? Why glucose split into 3 carbon molecules? Why not 4? Why not 5? Why ATP is invested before it's earned? Because we want to break these bonds, right? Right. So next video, I'm gonna talk about overall reaction itself, right? So have a nice time, I guess. Okay, let's meet you in next video. So this was glycolysis part 1. Next is part 2.